Yes, in the last lecture we have learned about the difference between the prokaryotic cell and the eukaryotic cell. The prokaryotic cell, it don't have the well-developed nucleus, and in the eukaryotic cells there is a well-developed nucleus. There is a defined nucleus. Eukaryotic cells, it has a membrane bound organelles. The prokaryotic cells, it don't have any membrane bound organelle. The prokaryotic cells, they are going to divide by the binary fission, and in the eukaryotic cells they are going to divide by the mitosis, meiosis. There is a sexual reproductions into the eukaryotic organisms. In the prokaryotes there is a asexual reproduction. Okay, so in this way there are the number of differences between the prokaryotic cell and the eukaryotic cell. When you look at the prokaryotic cell and the eukaryotic cells, in the both of them, okay, in the prokaryotic cell as well as in the eukaryotic cell, there is a presence of whom? There is a presence of the plasma membrane. Okay, though it is a prokaryotic, though it is a eukaryotic cell, what is present? The plasma membrane, it is present. And what is that plasma membrane? The plasma membrane, it is going to define the boundaries. Okay, what it is going to do? It is going to define the boundaries. Okay, that is, suppose we will consider here it is a eukaryotic cell. Okay, this it is an eukaryotic cell, and this it is a plasma membrane. That is the plasma membrane, it is the outermost covering of the eukaryotic cell. In the eukaryotic cell, which is an animal cell, in that there is the outermost covering, it is whom? It is a plasma membrane. Okay, so here it is the boundary. It is what it is going to do? It is going to define the boundaries. That is, it is going to separate out the internal environment of the cell and the outer environment of the cell. Okay, that it is defining the boundaries of the cell. It is going to it is going to separate out the cellular environment from the exterior environment. Okay, so this it is a plasma membrane. It is made up of the membrane. Okay, and here in the cell there are what? There are the membrane bound whom? There are the membrane bound organelles. Suppose here it is whom? It is what? It is the nucleus. Okay, it is a membrane bound organelle. Okay, then there is what? There is a endoplasmic reticulum. It is present. It is a membrane bound organelle. Then there are the Golgi bodies. They are present. Okay, there is the presence of whom? There is the presence of the Golgi bodies. There is a presence of the mitochondria. In the mitochondria there is a inner membrane. There is a outer membrane. So there are what? There are the number of membrane bound organelles. That is, there is a presence of what? There is a presence of the membrane. Okay, so the plasma membrane, it is the outermost covering which is going to differentiate between whom? It is going to differentiate which is going to separate out. Okay, which is going to separate out the interior environment of the cell from the exterior environment. Okay, now basically when you look at the plasma membrane, it is nothing but it is a lipid bilayer. What it is? The plasma membrane, it is what? It is a lipid bilayer, and it is consisting of what? It is consisting of the proteins. Okay, it is a lipid bilayer and it is consisting of the proteins. This lipid bilayer, when you say it is a lipid bilayer, so what type of a lipid it is consisting of? It is consisting of the phospholipids. What it is consisting of? It is consisting of the phospholipids. So here, okay, as we know, when you are going to say the lipids, it has what? It has a head and it has a tail. Okay, so this head, it is a hydrophilic. Okay, and the tail, it is hydrophobic. Okay, so this it is a long chain. Okay, this it is a long chain of the fatty acids, and here it is a it is what? It is a hydrophilic head. Okay, hydrophilic head, hydrophobic tail. Okay, and here at this hydrophilic head, who is present? There is a presence of what? There is a presence of these phosphates. So the lipid, it is what? The lipid, it is a phospholipid. Okay, lipid, it is a phospholipid. Okay, to the fatty acid there is what, there is a binding of whom, the phosphates, so that it is what, it is a phospholipid. So here, due to the phosphate there is a hydrophilic nature, and due to the long chain of the carbons it is going to give you the hydrophobic, hydrophobic environment. Now this, okay, this it is what, this is, it is a basically the phospholipid which is present. And now what it has been said, that lipid bilayer. Lipid bilayer means what? Okay, lipid bilayer means what? This it is an head, it is a tail. Okay, and this, okay, this type of an structure. This type of an structure it is called as what? This it is called as a lipid bilayer. Okay, bilayer, two layers. This it is a one layer. This it is a second layer. Two layers together it is what? It is a bilayer. So the membrane, the plasma membrane, it is consisting of the lipid bilayer, and in this lipid bilayer who is present? There is a presence of whom? There is a presence of whom? There is a presence of the proteins. So the plasma membrane, it is consisting of what? It is consisting of the lipid bilayer which is made up of the phospholipids, and it is having the embedded proteins in it. What it is having? It is having the embedded proteins in it. Okay, the proteins, it may be the integral proteins, it may be the transmembrane proteins, it may be the anchored one, or it may be the peripheral proteins. They are present where? They are present into this lipid bilayer, and this it is what? This it is a basic structure. Okay, this it is what? This it is a basic structure of your plasma membrane. Okay, are you getting? Now, the next one, it is what? The functions of the plasma membrane. What are the different types of the functions? Okay, what are the different types of the functions of the plasma membrane? First of all, it is going to define the boundaries. It is going to define boundaries. Second one, it is involved as, or it is work like as a selective barrier. Okay, next it is involved into the transport. Okay, fourth, it is involved into the cell signaling. Then it is involved into the cell-cell communication. Then it is involved into the cell adhesion. So in this way there are what? There are the number of functions. Okay, there are number of functions of whom? Functions of the plasma membrane. Okay, and it is also the center for. It is also the center for some of the reactions. Okay. So these are the number of functions of the plasma membrane. It is going to define the boundaries. Second one, it is a selective barrier. Now, when you look at the structure of the plasma membrane, okay, when you look at the structure of the plasma membrane, what you find? Okay, what you find that the nature. Okay, what you find that there is a lipid bilayer. Okay, and in that there is a presence of whom? There is a presence of the proteins. Okay, there are the presence of the proteins in this lipid bilayer. Okay, so here when you look at this it is what? This it is what? This it is a interior. Okay, this it is what? This is a interior of whom? It is a interior of the lipid bilayer. And when you look at this interior of the lipid bilayer, what you observe that its nature, it is a hydrophobic, because which region is there? The tail region of the phospholipids it is present. That's why the interior of, okay, interior of the plasma membrane, it is a hydrophobic. Now suppose this it is an outer environment and this it is an inner environment, and suppose here it is a molecule, and that molecule it is the hydrophilic, or it is a water soluble molecule. Okay, if that molecule it is a water soluble molecule, this molecule it is not going to be passed through whom? Passed through this plasma membrane. Why? Because as it is a water soluble, as it is a water loving, it cannot pass this hydrophobic region. Why hydrophobic region? Because the molecule is a hydrophilic, and here the interior it is what? Interior it is hydrophobic. So this molecule it cannot pass through whom? The hydrophobic region. That's why here there is a no easy transport, no easily transport of whom? The hydrophilic molecules which are present and in the outer environment to the inner environment. Means it is a barrier, means it is not allowing. Okay, it is a barrier, means it is not allowing. Okay, it is not allowing the molecules to get easily pass through who? The plasma membrane. Okay, are you getting? Okay, water soluble molecules or the ions, they cannot be passed through who? The plasma membrane. That's why it is going to be called as what? It is going to be called as an selective barrier. But if, okay, now if that molecule it is important for the cell and it has to be present, okay, it has to be present into the plasma, it has to be present into the cell, then at that time there has to be the intake of that molecule. For that purpose, it is allowing what? It is allowing the transport of the proteins, and here transport of the proteins or the transport of the molecules, and for that purpose the proteins which are present into the lipid bilayer, they are going to work. Okay, so the plasma membrane, not only it is a barrier, but it is also allowing the molecules which are essential for the cell through the system, and that system it is what? That system it is a transport system. There are the transporters. Okay, what are there? There are the transporters, and that transporters whose nature it is a protein, that transporter proteins, they are allowing the transport of whom? They are allowing the transport of the hydrophilic molecules. Okay, next here on the membrane, okay, there are the receptors. Okay, there are the receptors. That receptors, now they are going to accept the signal from the outer environment, and now they are going to respond to the outer environment. Thus the plasma membrane, it is also involved into the cell signaling, it is also involved into the cell communication, in the adhesion, and last one, it is what? It is a center for the reaction. Like how in the bacteria, the ETC, that is the electron transport chain, it is going to be carried out over the plasma membrane. Okay, so it is a reaction center. Okay, so the plasma membrane, it is going to provide the reaction center. So these are the functions of whom? These are the functions of the plasma membrane. It is going to define the boundaries, it is a selective barrier, it is involved into the transport, cell signaling, cell communication, cell adhesion, and it is involved, it is a center for the reactions, various biochemical reactions. Okay, have you understood the functions? Now we will go towards what? We will go towards the models of the plasma membrane. Okay, next we will go towards the models of the plasma membrane. It is the 1895 Overton, then there is a Gorter, and it is a Grandel in 1925, then in 1935, in the 1935 Daniel, it is Danieli and Delson, okay, in 1959 it is Robertson, and in 1972 it is the Singer and Nicholson. So first one, it is Overton. Okay, in 1895, he has said that the plasma membrane, it is made up of what? It is made up of the lipids. This it has been first suggested by whom? By the Overton. Then the Goiter and Grendel, both of them, they have postulated that the plasma membrane, it is a lipid bilayer. What it is? It is a lipid bilayer. Then the Daniel and Dawson, they have proposed the trilaminar model. Okay, trilaminar model of the membrane. Then the Robertson, he has given the concept of the unit membrane. And last one, it is a Singer and Nicholson. He has given, both of them, they have proposed the fluid mosaic model of a plasma membrane. Okay, so first Overton, he said that the plasma membrane, it is made up of the lipids, and Goiter and Grendel, they have experimentally they have shown that the plasma membrane, it is made up of whom? It is made up of the lipid bilayer. Danieli and Dawson, okay, they have proposed that the model, it is a trilaminar. Okay, the model, it is what? The model, it is a trilaminar, or you may say that it is a sandwich. Okay, what it is? It is a sandwich model. Okay, the sandwich model means what? Here, it is what? It is the lipid bilayer. Okay, and this lipid bilayer, it is covered by whom? It is covered by the proteins. So in between the two layers of the proteins, there is a presence of whom? There is a presence of the lipid bilayer. That's why it is a sandwich model, or it is a trilaminar model, which has been given by whom? Daniel and Dawson. Robertson, with the help of the experiment, okay, he said that, or with the help of the electron microscope, he has seen that there is a railway track like of the model. Okay, and here he proposed that the membrane, it is a unit membrane, that is, the membranes, they are similar to each and every cell. Okay, that is the lipid bilayer, it is not present into single type of an cell, but it is present over each and every type of an cell, that it is a unit membrane. Okay, by suggesting the unit membrane, he has also supported the model given by whom? Daniel and Dawson. Last one, it is a Singer and Nicholson. He has given the fluid mosaic model. Now there were the evidences that when there is a treatment of the phospholipases, okay, to the membrane, at that time there is a disintegration of the membrane. If the phospholipids, they are covered with the proteins, then there should not be the degradation of whom? There should not be the degradation of the lipids. But what has been happened? There is a degradation of the proteins, means the proteins, they are not covering whom? It is not covering the lipid bilayer. Then where the proteins, they are present? The proteins, they are present where? They are present in between, okay, or the proteins, they are embedded into the lipid bilayer. Okay, and there is a fluidity. Okay, there is a fluidity into the membrane. Okay, what is there? There is a fluidity into the membrane, and the mosaic structure means what? The various types of the proteins, they are present into the lipid bilayer. So that's why the model, it is a fluid mosaic model, which has been given by whom? Singer and Nicholson. Now on this there may be the question that which model it has been given by which scientist. Okay, suppose a fluid mosaic model, it has been given by who? Okay, so the number of options it has been given, you have to select the correct one. It is a Singer and a Nicholson. Okay, in this way there may be the question which has been asked into your exam. Okay, I think you may have clear up to this. In the next lecture we will learn about, okay, next lecture we will learn about the number of phospholipids which are present into the lipids, and also we will learn about the proteins which are present into the lipid bilayer, that is, which are present into the plasma membrane. Okay, so one more question from the today's topic that we have learned, one more question, it is what who is responsible for the selective barrier of the plasma membrane? So here what you have, what is the answer? The answer, it is a hydrophobic region. Okay, the interior of the plasma membrane, which is made up of the hydrophobic region, that it is responsible for the selective barrier. Okay, so we will meet in the next lecture.